Welcome to Medical Biochemistry 221. Today, we examine the specific metabolic failures of diabetic ketoacidosis, a condition where systemic tissues enter severe starvation despite a dangerous surplus of circulating blood glucose. This paradox is rooted in the cell membrane. Because glucose is a polar hydrophilic molecule, it is physically repelled by the hydrophobic lipid bilayer. To enter the cytoplasm and fuel metabolism, it requires specialized integral membrane glycoproteins to facilitate transport. This table outlines the tissue-specific distribution of these transporters. Tissues requiring a continuous fuel supply utilize insulin-insensitive channels. Red blood cells rely on GLUT1, and the liver utilizes the high-capacity GLUT2. These allow glucose to enter regardless of the organism's nutritional state. Cerebral neurons utilize GLUT3. This high-affinity transporter extracts glucose from the microenvironment, even during severe hypoglycemia, ensuring the brain remains fueled while other systems fluctuate. However, the body's largest metabolic sinks, skeletal muscle and white adipose tissue, possess no such autonomy. Without a specific hormonal signal, these tissues remain functionally impermeable to the energy supplies circulating just outside their membranes. These peripheral tissues rely on GLUT4. In the absence of insulin, these transporters are physically sequestered inside intracellular vesicles, keeping them away from the cell surface and preventing any significant glucose uptake. To mobilize these transporters, the cell must execute the signal transduction cascade detailed here. It begins as insulin binds to the extracellular alpha subunits of the receptor, triggering autophosphorylation of the beta subunits. This creates a docking site for insulin receptor substrate proteins, which activate the lipid kinase, PI3K. Activated PI3K then phosphorylates the membrane phospholipid PIP2 into PIP3. PIP3 acts as a recruitment signal for protein kinase B, or AKT, which is activated by PDK1. AKT then phosphorylates and inactivates the regulatory protein AS160. This allows Rab GTPases to transition into their active state, where they organize the cytoskeletal machinery to drive the exocytosis and fusion of GLUT4 vesicles with the plasma membrane. Without the completion of this exact molecular sequence, the glucose channels remain trapped inside the cell. In the context of diabetes, this represents a total mechanical failure of peripheral cellular feeding. The etiology of type 1 diabetes is a T-cell-mediated autoimmune destruction. As shown in the research material, the immune system synthesizes autoantibodies against specific pancreatic antigens, including glutamic acid decarboxylase 65, insulinoma-associated protein 2, and insulin itself. Once more than 90% of the beta cell mass is destroyed, the pancreas can no longer synthesize insulin. The brain detects an abundance of blood sugar, but because the GLUT4 translocation cascade cannot be triggered, the skeletal muscles and adipose tissues begin to starve. The body responds to this perceived starvation by mobilizing its finite liver glycogen stores, a first line of defense intended to maintain blood glucose levels during short-term fasting. These hepatic glycogen stores are highly limited. In a state of total insulin deficiency, the entire supply is completely depleted within approximately 18 hours. Crossing this 18-hour threshold forces the organism to abandon standard carbohydrate metabolism and activate the systemic, destructive fuel mobilization pathways that define a clinical crisis. In a healthy state, insulin actively inhibits the breakdown of white adipose tissue by activating PDE3B, which degrades cyclic AMP and keeps the lipid-mobilizing machinery dormant. Without insulin, this inhibition fails. Intracellular cyclic AMP remains high, causing unopposed activation of protein kinase A. It phosphorylates hormone-sensitive lipase and paralipin, triggering rapid hydrolysis of stored triacylglycerols. This causes a massive flood of free fatty acids and glycerol into the portal blood. Fatty acids undergo beta oxidation in the liver, yielding surplus acetyl-CoA. Simultaneously, aggressive gluconeogenesis drains the mitochondrial pool of oxaloacetate. Without oxaloacetate, acetyl-CoA cannot enter the citric acid cycle. Instead, this surplus is shunted into the ketogenic pathway, creating acidic ketone bodies, acetoacetate and beta-hydroxybutyrate. As these strong organic acids accumulate in the blood, they overwhelm the bicarbonate buffer capacity. The resulting drop in blood pH leads to severe metabolic acidosis and the deep, rapid Kussmaul respirations used by the body to blow off carbon dioxide in a desperate attempt to compensate. While the liver drives acidosis, a parallel crisis unfolds in the kidneys. The extreme concentration of extracellular glucose eventually overwhelms the sodium-glucose cotransporters responsible for reabsorption. This saturation point is defined as the tubular transport maximum for glucose. It represents the physical limit of the kidney's ability to pull glucose back into the bloodstream. Once blood glucose levels exceed approximately 180 milligrams per deciliter, these transporters are completely saturated. The excess glucose remains trapped within the renal tubules. This high luminal glucose concentration exerts a powerful osmotic pull. It aggressively extracts water out of the renal epithelial cells and prevents its reabsorption, leading to the excretion of massive volumes of urine. This process of osmotic diuresis is the direct cause of the profound systemic dehydration and polydipsia observed in diabetic ketoacidosis. We can unify these processes by tracing the biophysical loop. An absolute insulin deficiency triggers two parallel pathological pathways: uninhibited lipolysis and glucosuria. Following the flowchart, lipolysis leads directly to ketonemia and metabolic acidosis, while glucosuria drives osmotic diuresis, resulting in life-threatening dehydration. This explains the clinical triad of polyuria, polydipsia, and hyperphagia. However, the patient's experience of extreme compulsive hunger while their blood is saturated with fuel reveals a critical nuance in glucose transport. It is a common mistake to assume the entire brain is starving. In reality, cerebral cortex neurons utilize the insulin-insensitive GLUT3 transporter. They continue to import glucose freely, even in the total absence of insulin. The hunger reflex is triggered specifically by the cells of the hypothalamic satiety center. These cells utilize the insulin-dependent GLUT4 transporter. While the rest of the brain is fueled, these specific sensors are starved, causing them to fire the continuous hunger signals that drive hyperphagia. Diabetic ketoacidosis is the catastrophic collision of two unregulated starvation responses: acidosis and dehydration. This systemic collapse dismantles homeostatic balance until insulin is medically restored.